components that typically have receptor sites on them that receive information. Dendrites. Dendrites can receive information from other cells. What other parts can? Cell bodies. Yep. Less common, but yes, cell bodies can receive information directly. So the little area of transfer of information, what do we call that little area there? The synapse. Yep. So some textbooks will specify synaptic cleft or synaptic gap. Just way easier to say synapse. Okay, so a lot of information going on in the synapse. You should have in your notes neuron A and neuron B. Remember that from last week? Can you guys visualize that? So when neuron A, when the dendrites take in the information, that information, once it's in the dendrites, is in electrical form. Okay, so the dendrites take in the neurotransmitters, which are chemicals, but once they've got the information, now within the neuron, information is transmitted via electrical format. Okay, so outside the neuron, once the information is spit out from the axon terminals of a neuron and is spit out into that little space, in what format is the information? Now it's chemical. What do we call those chemicals? So we're going to spend a whole lot of time right now on talking about what goes on within the neuron. Once we're done with that, after the first or second break, then we'll talk a little bit more about how information gets transmitted between neurons. But right now, we're really going to focus in on what goes on inside the cell. So the axon hillock, if you recall, is the structure that is unmyelinated, and that is where action potentials kind of arise from. So if you had to describe to someone what the axon hillock is, what would you say? The action potential generator, that is a great start. Neurons are three-dimensional cells. You guys know that. Okay. Turns out that there's fluid inside a neuron. There's also fluid outside of a neuron. Are you with me so far? There are specific ions, which are little charged particles, both inside and outside that fluid. So we have fluid inside the neuron, which we call intracellular fluid, because it's inside the cell. And the intracellular fluid has a particular mix of ions, some positively charged, some negatively charged. Are you with me so far? The fluid outside the neuron, we call the extracellular fluid, outside the cell. That fluid also has a particular mix of ions, some positively charged, some negatively charged. All right, with me so far? Okay. So it turns out that the ions, and we'll discuss some of the major ones, the ion concentration, both outside and inside, always have to be separated. The cell membrane, the neuron membrane, or the neural membrane, is really essential in separating the intracellular fluid from the extracellular fluid. The neural membrane is really important in separating, or kind of keeping, the intracellular fluid in, the extracellular fluid out. If something were to go wrong with that neural membrane, it's really going to screw up the entire process of how this neuron is able to send information. So this axon hillock is going to generate an action potential, but it can only do that if there's the right concentration of ions both inside the cell and outside the cell. Okay, so the neural membrane, oh my gosh, it's really, really important that it does its job. The membrane is permeable. What does permeable mean? Yes, some particles are able to pass through to an extent. It's not like any ion or particle can pass through, but some are allowed to. All right, so do you guys see how this membrane is kind of basically a lipid layer, and there are some little passageways embedded within that membrane. Some ions are able to flow in and out through these little channels. Are you with me so far? Okay. So when we say that the membrane is somewhat permeable, those channels give it a little bit of permeability. Ions that we're going to talk a lot about. Okay, ready? Sodium. Sodium is a big player. You guys remember the element abbreviation for sodium? Sodium is Na. How about chloride? How about potassium? Those are the three major ones. We're going to talk about calcium as well. So now, just This line we have 4 major ones. Inside the cell, there is a specific combination of ions. Outside the cell, there's a specific combination of ions. The separator of the two would be the neural membrane or the cell membrane. The only way that we could get an action potential to generate is if there happens to be a huge influx of sodium into the cell. That is the only way we can get action potentials to start going, to trigger. Okay? So in order for us to have like a huge influx of sodium into the intracellular fluid, where does most of the sodium have to be? Probably on the outside. The only way sodium can rush into anywhere is if there's a ton on the outside and not so much on the inside. Does that make sense to you? I've used the term action potential a lot. Action potential is the development of an electrical signal, and it's how information is moved or transmitted within the neuron. Let's get that down. So again, action potentials arise from the axon hillock. The only way an axon hillock could trigger an action potential is to get a bunch of sodium inside the cell. Hold off on the sodium for a second. Let's just say a bunch of sodium gets in the cell like we want it to. So the axon hillock is like, whoop, huge rush of sodium, we're good, go ahead and light the fire or trigger the action potential. So the action potential will then start to move down the length of the axon. You guys know from last week what happens once it reaches the ends of the axon here, the little terminals with the buttons.
Turns on neurotransmitters.
Yes, neurotransmitters will then be released. Are you with me so far? So our job today is to figure out how this axon hillock triggers an action potential, how it moves, and you already know the neurotransmitters in the synapse part. Okay. All right, this image is from your textbook. So what this is showing, this is the neural membrane. Okay, so this is the neural membrane. This is the intracellular fluid. This is what's inside the cell. This is the extracellular fluid. This is what's outside the cell. Are you with me? Okay. So what they're also showing is embedded within the neuromembrane are some channels or pumps or some ways that ions could get out or in. So let's talk about what's embedded within the neuromembrane for a moment. So, if I were you, I would jot this down. I mentioned to you that neural membranes are permeable to an extent, right? Okay? They have to be permeable to an extent if we ever want sodium to rush into the cell. So the question researchers had is what causes or what controls that permeability of the membrane? And here are your two answers. One of them would be ion channels embedded. These are protein structures embedded within the neuron's membrane. Allow certain ions in and allow certain ions out so they can act as gatekeepers. One type, voltage-dependent ion channels, respond to nearby electrical signaling. Then our second type of ion channel, and the other type of ion channel would be what we call a ligand-gated ion channel. So ligand respond to chemical messengers. So again, what we're saying is around every neuron is a neural membrane. Neural membranes are permeable to a particular degree. How are they permeable? Well, some of them, along the way, we have these ion channels. These are little gatekeepers of the neural membrane. And they are ion selective. Means only responds to certain ions. So if we happen to have an ion channel that's specific to potassium, chloride and sodium aren't really going to be able to get in there because it's specific only to control the permeability of the cell membrane. Two things control permeability. We just talked about the ion channels. The other thing that controls permeability would be the ion pumps. Have you guys in your bio days heard of a sodium-potassium pump? Yes. Okay, so that's one of the pumps we're going to talk about. So backing up, around every neuron is a neural membrane. Certain ions need to get in, certain ions need to stay out. Okay, how do they get in and out? Well, the permeability of the membrane is controlled by the channels and the pumps. Are you with me so far? Channels basically let ions move freely in and out. Pumps require energy. They specifically have to go in there and get ions out. Okay, there needs to be the right amount of ions both outside and inside to allow that action potential to be triggered. Interesting to make note of is that 20 to 40% of the brain's energy is used on these pumps. Okay, when a neuron is at rest, there is more sodium outside of the neuron. Also, when it is at rest, there is a lot of potassium inside of the cell. When you have molecules of high concentration, they naturally want to move to where there's not so many of them. It's just what happens. So that movement we can call diffusion. They naturally want to redistribute so it's equal both inside and outside of the car. Does that make sense to you? So the one term that you guys want to know is diffusion. Why do they move? Well, when there's a concentration gradient, which means when it's higher in one area and lower in one area, they're going to move to make it equal. If there's a lot of sodium outside the cell and very little inside, do we have a concentration gradient?
Yes.
Yes, we do. We have way more sodium outside the cell. Not very much inside the cell. A concentration gradient exists. So what's going to happen just as soon as there's some openings for sodium? As soon as there's an opening, it's going to rush in. Diffusion is going to happen. The only way an action potential can be triggered is if sodium rushes into the cell. At rest, the neuron's potential is about negative 70 millivolts. It's called it the rest potential. Depolarization is to become less negative and moving towards zero. So this dotted line is the threshold. I have it here at negative 55. The minimum amount of input needed for something to actually happen. If enough gets into the cell and we're adding all these positive ions, we're going to move away from negative 70 to negative 60 to negative 50. And once we get here, do you see where we have met or exceeded our threshold? That's the minimum amount we needed. Just as soon as you reach or exceed that threshold, an action potential happens. At some point, the sodium channels have to close. That's their job. They open, they close. Also, what happens after, at some point, the potassium channels have to open. What's going to happen when the potassium channel opens? Potassium is going to fly out. Why? Why is potassium going to leave? Diffusion, because there was a concentration gradient. Are you with me? Okay. And then eventually potassium gates are going to close. So there's this process of sodium opening, sodium closing, potassium opening, potassium closing. This has to happen in the right order. Enough of it has to move from inside the cell or outside the cell in order for an action potential to occur. So here's